at the back end. Let me just give them the heads up that we are ready to go.
Okay, yeah, in now.
Okay, that's fine. Hello, everyone. Hello, good day, good morning, good afternoon, from wherever you are joining us from. We are so happy to have you all here. Thank you so much for joining us, the African Vaccine Academy and the Merck team on the call and our distinguished speaker today. So I am Oluton Sanusi Oladuni and the lead of the African Vaccine Academy at Bloom Public Health. I am so happy to have you all join us. At Bloom Public Health, our mission is deeply rooted in building resilient public health systems and ensuring equitable access to health solutions across Africa. We truly believe that fostering knowledge and collaboration is paramount in achieving this vision, and this webinar is a testament to that commitment. The journey of a vaccine from initial research stages to becoming a vital tool in protecting our communities is a complex and meticulously regulated process. We all of us on the call understand the process we need to go through to get it done and to get it completed. Understanding this critical role of clinical and preclinical trials is essential for all stakeholders. We all here are stakeholders, even if you're working directly in the lab or you have kids or you have colleagues or even you yourself, you've actually gone through the process of immunization or vaccination at one point in time or the other. So we are all stakeholders in public health and the vaccine ecosystem. It's these rigorous phases that guarantee the safety and effectiveness of the vaccine we rely on to safeguard our collective future. So today, the African Vaccine Academy is partnering with Merck in this series, a global leader. Merck is really a global leader. You cannot mention Merck and not mention vaccines. You cannot mention Merck and not mention so many other things that they do in the healthcare space. They are really a driving force in vaccine innovation. Their dedication to scientific research and public health aligns perfectly with our own mission in Bloom Public Health. And we are thrilled to bring you their expertise through this session. We understand that a lot of esteemed guests on the call, and we acknowledge each and every one of you. We appreciate you for joining us today. And now it is my pleasure to introduce our esteemed speaker for today, Dr. Anita Ben Taylor. Before she goes on, let me just give us a brief on her extremely impressive profile. Dr. Taylor is a biopharmaceutical expert and holds a PhD in biochemical immunopharmacology from the University of London and a postdoctoral fellowship focusing on preclinical models, elucidating biological mechanisms in cardiovascular health. Our expertise further extends through faculty. Our expertise further extends through faculty positions at renowned institutions, including Johns Hopkins and the National Institute of Health, where she focused on critical research into vasculopathies associated with sickle cell disease. Currently, Dr. Taylor brings her deep scientific knowledge to the global biological technical marketing team at Millipore Sigma. Dr. Taylor, we are absolutely delighted to have you share your wealth of knowledge and experience with us today. Thank you for being part of this crucial webinar. Over to you, Dr. Taylor, for your very insightful presentation. We can see the slides already. Over to you, Dr. Taylor.
Great. Thank you very much for the introduction. And I also want to thank everybody. Firstly, I want to thank Merck for hosting this, but I also want to thank, the organizers on the South Africa side who have just worked diligently to get this all together for you guys, for the Vaccine Institute. So it is my pleasure. A country one day I hope to visit. But it is my pleasure to present to you some of the learnings that are happening in the vaccine world, just in particular the mRNA vaccine, and how people are starting to implement and some of the rules and regulations around it. I may have to go on camera because it's going to start flickering shortly, so I'll just get off camera for a bit.
It's fine.
And come back.
It's fine.
Fantastic. So I will kick it off, and if you can't, if the volume goes out, please do let me know. So yes, thank you. So today I'm going to talk about safeguarding the future and the role of preclinical and clinical trials in vaccine development. I'm really going to navigate this process of what researchers are doing and how that leads into clinical phase trials. And hopefully it'll move. Perfect. So this is the agenda. So I'll give you a high-level overview of the introduction to vaccines, what preclinical studies are performed in various models to move forward, how then you transition into clinical studies, and then really, as a company, as Merck, how are we providing solutions to enable our researchers? And so I'm going to start off with the vaccine timeline. I think, vaccines have been around for a very, very long time. And it's really transformed global health and saved millions and millions of lives in combating infectious diseases. And I think we all know, it all began with Edward Jenner in 1796, who created the first vaccine against smallpox. And this really laid the foundation for immunology and how the immune system works in combating infectious diseases. Moving forward to 1890, Emil von Behring followed with the vaccine, tetanus vaccine, which advanced the ability to prevent deadly infections. And as you move into the 20th century, key vaccines started to emerge as preventative measures, such as the tuberculosis vaccine in 1921, the first influenza vaccine in 1937. And then you have the introduction of the vaccines for polio, measles, mumps, rubella, all things that I think we're all familiar with, the routine MMR vaccines. And this really led to the significant public health achievements as far as preventative preventative measures being taken. And it also included the US becoming polio-free in 1979 and the eradication of smallpox in 1980. And as you move through to the 21st century, we saw the development of the HPV vaccine in 2006, and more recently, we've seen the COVID-19, the rapid generation of COVID-19 vaccines in 2020, which we all experienced. And this really showcases the ability to respond to emerging health threats and the critical role vaccines play in saving lives and improving health outcomes worldwide. So what is it that we're looking at? What are we trying to do with an immune response? So when an individual is vaccinated, it triggers an immune response. And initially, you'll have a primary response. And that's really designed to protect you by producing antibodies. And the primary response, it's relatively slow, typically peaking around about 7 days after exposure. So it's not a very strong response, but during this time, what your body is trying to do is generate antibodies, what we call memory B cells, so that they can confer long-term immunity should you be exposed to the same antigen at a later time point. And so when the body encounters the same antigen, the immune system responds much faster and more robustly. And due to the memory B cells, production really kicks in very, very quickly. So the titers will be a lot higher, producing much more stronger and more effective protection against reinfection. So what are you trying to achieve with vaccination? And I think we all experienced this. We all heard about it, during the COVID-19 pandemic. Vaccination was a key tool, and it still is a key tool in any infectious disease in achieving herd immunity. And it's a state where enough people in the community are immune either through vaccination, reducing the virus's ability to spread. And the threshold for herd immunity varies by disease depending on the RO value. And that's really about how many people on average one infected person can spread the virus to. And so as we all experienced and heard for COVID-19, it was estimated that around 80% of the population needed immunity to protect those who unfortunately couldn't get vaccinated due to medical conditions or other conditions and to slow transmission overall. Now, as I said, and I think you saw earlier on the first slide, vaccines have been around a long, long time, and they've come a long way over the centuries. And there are multiple different types of vaccines, from early live attenuated and inactivated vaccine to cutting edge mRNA and viral technologies. And as infectious diseases continue to evolve, so do the tools that are used to fight them. And so thanks to vaccines, diseases like measles, polio, and influenza have been dramatically reduced. And more recently, mRNA vaccines have been kind of the game changers especially during the COVID-19, where it really brought it to the forefront as a technology, proving their potential to be rapidly developed and be highly effective at the same time. And this success has really opened the doors to future breakthroughs, including next generation mRNA vaccines for flu, RSV, and even personalized medicines such as cancer immunotherapies. However, despite this innovation, one of the most crucial things is accessibility, making vaccines accessible to everyone. Manufacturing and distribution plays a huge role in making sure vaccines reach those who need them the most. So efforts are being made of global partnerships between industries and governments, which are important for ensuring equitable vaccine access. And one of the biggest lessons that was learned from the recent pandemic is being vaccine ready. New infectious diseases are continuing to emerge, and I think we're already starting to hear about all these outbreaks such as RSV and even measles as well. And with technology such as mRNA and other rapid deployment technologies, we can respond a lot faster and stay ahead of our future health threats. And furthermore, I really want to emphasize here, regional production is key as well to ensuring a reliable supply and strengthening the preparedness for future outbreaks. And especially in South Africa, 40% of the vaccines will soon be manufactured in South Africa. Now, with all that being said, let's have a look at the regulations. To the point that was made earlier, it's an important area to understand and how it plays into vaccine development. Vaccines have been the cornerstone of public health. However, the regulatory oversight has really lagged behind therapeutics and biologics, and historically the process lacks clear guidelines. And it really has been a bit of a wild, wild west as far as vaccines are concerned and the development of vaccines. And I think the COVID-19 pandemic really changed everything. Can it really change the way that regulators are looking at vaccines? And one of the things that, in some of the meetings that I've attended, it's about global collaboration and approvals that really are defining how vaccines are going to be developed. And COVID-19 really led that way. So agencies like the FDA and EMA are working to build on these frameworks, creating a more structured and predictable vaccine approval process similar to biologics and therapeutics. Now, mRNA technology itself, it's not new. So it's been around for many, many, many years. And as a result of COVID-19, it has really come to the forefront. And it really goes beyond vaccines, paving the way for precision medicine, immunotherapy, and breakthrough treatments. Now, we've already seen how mRNA vaccines are being successfully implemented for infectious diseases like COVID-19, but the potential doesn't really stop there. And you're getting development of mRNA cancer-based vaccines that train the immune system to recognize and attack tumor cells, providing personalized cancer treatment. Beyond vaccines, it's also revolutionizing cell therapy, such as CAR T therapy. For example, it can help enhance immune cells to fight cancer more effectively. It also is being explored for regenerative medicine, for targeted therapies in heart disease, blood disorders, and so forth. And it's also making waves in therapeutic protein production, where a patient's own cells produce the protein needed for treatment. And these have applications in things like immunotherapy, where mRNA can be used to enhance the immune response against diseases such as cancer, encoding proteins that stimulate the immune system to recognize and attack its own cells. Gene editing, I think we've seen the success of sickle cell, treatment for sickle cell, where you're using gene editing tools such as Cas9 to enable precise genetic modifications, and protein replacements, restoring missing or faulty proteins in genetic disorders such as cystic fibrosis and metabolic diseases, where the body lacks specific enzymes or proteins. But for today, with all that in mind, for today, even though there are different types of vaccines and the way that they are being used, today's presentation will really just solely focus on mRNA vaccines and infectious diseases. So what is an mRNA vaccine? So mRNA vaccines typically just instruct cells to produce a harmless viral protein, which triggers an immune response. And typically, the mRNA is encapsulated within lipids or carriers such as lipid nanoparticles or polymers to protect it from degradation and facilitate cellular entry via endocytosis. Once inside the cell, the LNPs destabilize and release the mRNA into the cytosol. Here, the ribosomes translate the mRNA into the viral protein, and the newly synthesized viral protein is processed and presented on the cell surface by the major histocompatibility complex 1. And this is crucial for the activation of the immune system. Antigen-presenting cells, such as dendritic cells, take up the protein and process it and present it to T helper cells, or CD4 cells, via MHC class 2 molecules, which activates the T helper cells. And these, in turn, stimulate these cells to differentiate into plasma cells, which end up producing antibodies. And these are the antibodies that are specific to the viral protein and can neutralize the virus by binding to it, preventing it from entering host cells and infecting. So, and during that process, some of the activated B cells become memory B cells, which persist in the body and provide long-term immunity. And these cells are there primarily, as explained earlier, to quickly respond to future exposures or rapidly producing antibodies. So the development of mRNA vaccines follows a pretty similar process to large molecules, biologics, therapeutics, but it's a little bit more streamlined and linear. It's actually a lot more easier to develop. And mRNA vaccines are actually more safer compared to traditional biologics because they tend to be a little bit more complex. And the journey really begins with the exploratory and preclinical phase, where mRNA is synthesized and tested in lab models, which will be both in vivo and in vitro, to evaluate safety and effectiveness. And once preclinical results are promising, regulatory approval is obtained to begin human trials following the standard phases of phase 1, phase 2, and phase 3, which I'll go into a little bit more detail later. If successful, the vaccine is then manufactured at scale and made available to the public. Yet it is continually monitored for safety and efficacy. And this entire process can typically take anywhere between 10 to 15 years. Currently, there are over 100 mRNA vaccines active in clinical trials, and with a lot more in preclinical development as well. And so I'm going to move on to the next section, which will cover the preclinical studies. And so this is just kind of a bit of a reminder. So developing mRNA-based therapeutics and vaccines, it's a very structured process. It's a step-by-step process to ensure safety and efficacy, as well as scalability. What you're seeing here is really a high-level overview of the mRNA development process. Whether for infectious diseases, cancer, or genetic diseases, the first step is really understanding the molecular mechanism and immune responses needed to design an effective mRNA therapy. Once the target is identified, the mRNA sequence is designed that encodes the specific antigen or therapeutic protein needed to trigger an immune response or correct a disease-related dysfunction. The designed mRNA is synthesized to create a functional mRNA, and the goal is really for high purity and scalability, but for large-scale production. Now, since you want to deliver the mRNA into the host by itself, cellular uptake efficiency of naked mRNA is extremely low. And one of the reasons of this poor uptake is really because mRNA is negatively charged and it is repelled by the cell membrane, the anionic cell membrane. And secondly, mRNA by itself rapidly degrades due to extracellular RNases. And so to overcome some of these obstacles, different strategies are used for mRNA delivery, for safe and efficient delivery. So it's typically encapsulated in molecules such as LNPs or lipid nanoparticles, which are cationic in nature, or others such as polymers and peptides. And before human trials can begin, mRNA therapies will undergo preclinical phase, where you're doing testing in both in vitro and in vivo for safety. Then it's manufactured, and then it will go into large-scale first-in-human studies. And so today's focus will be the preclinical side of things, and I will also go into the clinical side and what is required for that. And so what are we looking for? The journey really begins with preclinical, in vitro, and in vivo animal studies. And this is crucial in understanding how the candidate vaccine interacts with biological systems. And the studies focus primarily on safety, that's the big component of it, immunogenicity, and efficacy. Is it doing what it's supposed to do? Now, I want to preface this because a lot of preclinical work is being done to assess mRNA vaccines. And I want to highlight the importance of how biological models are used for translational research, whether it's therapeutics or mRNA. Developing safe and effective mRNA therapeutics requires advanced models that closely mimic human biology. And while traditional 2D cultures are valuable for early stage screening, assessing mRNA expression, cytotoxicity, and immune response, they often lack the complexity needed to fully predict the in vivo outcome. And to improve this and reduce clinical trial failures, we're seeing an increase in an integration of tools such as 3D tools, such as organoids and spheroids, as intermediary before you go into in vivo models to enable better prediction. And so 3D models essentially, if you're not aware, they provide a more physiological relevant system by recapitulating the complexity of actual organs and provide a more accurate and biologically relevant platform for investigations compared to 2D cell cultures. And it helps in terms of replicating tissue responses, immune interactions, and mRNA delivery efficiencies, providing better insights into how mRNA therapies behave in a human-like environment. And this really is bridging the gap between early screening and clinical translation. And then, of course, you are going to move into in vivo models, which are essential for validating biodistribution, toxicity, and long-term efficacy to ensure that the mRNA vaccine is safe and effective before it goes into clinical trials. And I do want to highlight here, as Merck as a company, we are partnered with vendors such as BioVT, which provides high-quality immune cell subsets and biologically relevant 2D models to optimize that early stage mRNA screening. And we've also acquired hub organoids, which offer patient-derived organoids and iPSC-derived organoids, allowing researchers to study mRNA therapies in a more human-like system. And so by combining both 2D, 3D, and in vivo, you're building a more predictive, scalable, and having a more robust approach to mRNA drug development, bridging that gap between preclinical discovery and a clinical successful outcome. So one of the initial steps you're going to be doing right at the beginning after generating an mRNA, when you've got your libraries, is selecting the right mRNA candidates. It's a critical step in developing effective vaccines. And this process involves screening and validation stages, functional validation stages, to ensure the selected mRNAs are well expressed and functionally relevant. And so the first step is to determine which mRNAs are actively expressed within the system. There's tools such as single-cell RNA-seq sequencing. This is a really powerful tool. It's a tool that's becoming very prevalent in research, and they are used to analyze the expression profiles of individual cells, and it allows you to examine the transcriptome at a single level. And so you're seeing the identification of cellular heterogeneity, can never say that word, within a tissue or a sample, revealing the distinct cell types. And multi-seq is really our offering as a high throughput version of this, so you can literally have more bang for your buck, boosting efficiency and accuracy. There's also techniques such as qPCR, RT-qPCR as well. And this essentially validates expression levels of identified mRNA candidates in samples. And then also fluorescently tagged mRNAs that are either tagged with GFP or RFP, which enables visualization of mRNA expression in biologically relevant cells. Now, once you've identified your promising candidate, you need to make sure it's doing what it's supposed to do, its biological effects. And it's tested both through cell-based assays and in vivo models. Cell-based assays, as I said earlier on, typically can be 2D or 3D models, that are biologically relevant to what you're looking at to mimic those conditions. You will, since you're working with cells, you'll have cell health assays that you will want to ensure that you're assessing the viability and their health, such as NTT assays, LDH assays. And then ELISA and flow cytometry tools are also used to confirm activation and protein expression. Animal models are also used, so using relevant animal models. Non-human primates are probably one of the most common models because they do mimic the human structure or architecture, and then mice are often commonly used. And what you're doing is you're really kind of evaluating immunogenicity, i.e., is it invoking immune response? And then its therapeutic efficacy as well. And you're also looking to see how it's behaving within the system as well. With the advent of CRISPR, these tools are also used to genetically modify cell lines or animal models to assess mRNA vaccine efficacy and safety. By turning off genes on or off, you can see how it affects gene expression, immune responses, are there any off-target effects, and really the overall vaccine effectiveness. And this really helps pinpoint ways in which you can improve vaccine efficacy and safety whilst uncovering the mechanisms behind immune protection. So this is just an example of a case study that we have that really demonstrates 3D models, patient-derived organoids, how they can be used to evaluate LNP mRNA encapsulated with LNP payload delivery in a biologically relevant model for preclinical research. And here the goal was to assess whether this can be successfully delivered into a complex system to confirm functional uptake at the cellular level. And to do this, we used GFP mRNA encapsulated in LNPs. It was introduced to these PDOs via electroporation. The cells were then incubated and analyzed at two time points, 5 hours and 22, using fluorescence microscopy. And at 5 hours, as you can see, there is limited fluorescence, but at 22 hours, you saw the GFP expression, which was clearly visible, confirming that the mRNA had been delivered and translated into the protein. And then to further quantify this, flow cytometry analysis was performed, which showed a 96.6% GFP positive population in these LNP-treated organoids compared to the zero control. And this really provides strong evidence that LNPs are being efficiently delivered and they are functional in patient-derived organoids, leading to measurable expression. And so this study really highlights how organoids can be used and be used as a powerful tool to evaluate mRNA delivery and payload functionality in a controlled physiological relevant system. Now, another important aspect, and probably one of the most important aspects in looking at how vaccines work, is looking at it both the quantity and the quality of the immune response to these vaccines by looking at the humoral and T-cell mediated immune responses. So taking you back to your immunology days, and how effective and durable they are. And so unlike traditional vaccines, as you will know, mRNA vaccines work by instructing cells to produce a specific antigen, which then triggers both the B and T cells. And B cells generate antibodies in a T-cell dependent manner so that they can recognize and neutralize the virus. And by measuring these antibody responses, you can tell how well the vaccine is stimulating humoral immunity. And often the most common methods that are used for measuring this are usually typically ELISAs, which are single-plex assays. But to get more bang for your buck and more higher throughput, you have the advantage of the multiplex assays that allows you to do simultaneous detection of multiple antibody isotypes, whether it's IgG, IgA or IgM, depending on based on what serotype that is embedded on the bead. And it provides a much more complete picture of the immune response over time and which of these isotypes are prevalent. But how do you know that your vaccine is doing what it's supposed to do? And so while serology assays tell us how many antibodies are produced, you need to know if they are working. And neutralizing assays, and sometimes they're called microneutralizing assays, assess whether antibodies can block the viral entry into the host cells, preventing infection. And in this test, what you're doing is you're taking vaccinated serum, mix it with the virus, titrate, and if the antibodies successfully prevent virus-host interaction, it confirms a strong protective immune response, and you'll actually have a visualization of that. Now, we know that the immune system also isn't about generating antibodies. You've also got T-cell immunity against vaccines as well. And these play an equally important role in providing long-lasting protection. So mRNA vaccines, as I said earlier on, your CD4 positive helper T cells support B cell activation and production. Your CD8 cytotoxic T cells directly eliminate infected cells, preventing the viral spread. And to fully understand the strength and the durability of the immune response, you use a combination of molecular and immunological assays. So things like flow cytometry, what you're doing here is you're really characterizing your CD4 cells and your CD8 cells, because you want to make sure that these are being activated, which will ultimately lead to B cell formation. And so that's just a measure of activation, that your immune system's doing what it's supposed to do, and you're getting the generation of markers, and then also the generation of B cells, and then